Auzubillahiminashaitanirrajim. Bismillahirrahmanirrahim. Today our topic is about neuromuscular junction. What is neuromuscular junction? Neuromuscular junction is actually the junction for the transmission of electrical impulses from the neuron ending to the muscles. And we can say how the muscles show response to the nerve impulses. Today our topic is neuromuscular junction, or we can say neuromuscular transmission. Neuromuscular transmission. Depolarization will come on the membrane of the neurons, and it will release some special types of chemicals. These chemicals will bind on the end plate, and here it will produce depolarization. So simply we can say how the muscles contract, when there is depolarization comes from the neuron ending. So today our topic is neuromuscular junction. Neuromuscular junction is actually the meet point between the terminal of the neuron and the end plate of the muscles. This meeting point is called neuromuscular junction. Or we can say the junction or the gap between the presynaptic vesicle or presynaptic membrane and postsynaptic membrane is called synaptic cleft. Neuromuscular junction is also called neuromuscular synapses. This is actually the meeting point between the neuron ending and the muscle end plate. In the previous lecture, we have studied that how neuron talks to each other. In this lecture, we will study how neuron talks to the muscle, due to which the muscle shows contraction phenomena, or we can say due to which the muscle shows movement. So, how neuron talks to the muscle, today we will study this. Neuromuscular junction is simply like an ideal couple in which the wife always talk and the husband always keeps on listening. Example of the nerve terminal, this is like wife which always talks, and end plate is the example of husband which always keeps on listening. So this is how the neuromuscular junction talks to each other, like the wife talk to his husband. So now we will study the neuromuscular junction in detail. How muscles show contraction, or how the body muscles show movement to the action potential. Suppose action potential, this is the neuron cell body, this is actually axon, and this is neuron terminal, or we can say nerves terminal or nerves end. So the action potential is going over the membrane of what? Over the membrane of the axon, and it reach into the nerves terminal. As you know, there is sodium channels. Which are voltage regulated sodium channels. So as the action potential reach from the previous lecture, there will be a huge influx of what? Sodium ions. So these sodium ions will stimulate another sodium channels, and the depolarization will increase. So because of this depolarization, the inside electronegativity will decrease and the electropositivity will increase. There is a special types of channels which are called depolarization sensitive channels over the membrane of the axon terminal. These channels are called depolarization sensitive calcium channels. These are what? These are depolarization calcium sensitive channels. So when the depolarization reach, there will be a huge influx of which ions? Calcium ions. Why? Because there is a huge concentration of the calcium ions in the extracellular environment. So it will move against the concentration gradient into the intracellular environment. Now what will happen? Neurotransmitter will release. But yet we haven't studied that how neurotransmitter synthesize. How neurotransmitter synthesize. Remember, neurotransmitter are bounded by a special types of proteinaceous vesicle, and these proteins are not synthesized in the nerve terminal. Remember, these proteins are not synthesized in the membrane terminals. From where it will come? It will come from the neuron cell body, because neuron cell body has DNA. DNA will transcript into messenger RNA. Messenger RNA will translate into proteins, and these proteins will transfer into the nerve end. Now, as you know, neurons ending has mitochondria. Suppose there is mitochondria. So mitochondria, it will synthesize what? Acetylcholine. What? Acetyl coenzyme A. The mitochondria release the acetyl coenzyme A, but there is no choline inside the neuron ending. There is no choline inside the neuron ending. So there is a special types of transporter present over the membrane of the nerve ending. This transporter is called choline transferase. What? Choline transferase, and these are also proteinaceous. And these proteins also come from the neuron cell body. These choline transferase, it will capture the choline which are not synthesized over there. It will be captured from what? From the GI tract. From the gastrointestinal tract. The choline will be transferred into the intracellular environment by the choline transferase enzymes. So this choline will what? It will combine with this acetyl coenzyme A will convert into acetate. What? Acetate, the coenzyme A will release. This acetate will combine with choline and it will form what? Acetyl. The neurotransmitter, it will release into the extracellular environment by the exocytosis process. Now there is a special types of receptor. Or we can say channels. There are channels. And into these channels there are receptor sites, and these receptor sites, these neurotransmitter will bind. What will bind? Neurotransmitter will bind. This channel is actually what? This is protein. Which type of protein? This is pentamerase protein. Pentamerase means these are composed of 5 types of proteins. 2 alpha, 1 beta, 1 gamma and 1 delta. This is called pentamerase channels. Pentamerase channels. Now it will bind into the receptor site. As you know, there is sodium ions. So the sodium ions will move into the intracellular environment. But there is not much sodium to lead the resting membrane potential into the threshold potential. Remember, there is not much sodium influx that will lead the resting membrane potential into the threshold potential. So we can say it will lead the resting membrane potential from -70 to 0. From -70 to 0, we can say such type of potential which cannot lead into the depolarization or which cannot lead into the action potential. This is called end plate potential. What? Motor end plate potential, or we can say simply end plate potential. Now this end plate potential depends on the concentration of the neurotransmitter bind into what? Into the receptor site. If the neurotransmitter release in a large quantity and bind to the receptor site in a large quantity, then the end plate potential will be greater. But suppose if there is 1 or 2 molecules of or chemicals of the neurotransmitter, there will be a little increase, suppose 0.4 millivolt. 0.4 millivolt increase in the potential. So such type of small increase in potential is called miniature what? Miniature end plate potential. Such type of potential is called miniature in-plate potential. Miniature means minute, small amount. When there is small amount of neurotransmitter release. Now remember, the calcium which enter inside through a special types of channels, and also remember one point, these are not voltage regulated channels. These are not voltage regulated channels, but these are sensitive to chemicals. These are sensitive to neurotransmitters. So we call this channel is ligands channel. What? Ligands channel. So through the ligands channel, the sodium influx will occur. This sodium ion, it will move into where? Into the neighbor membrane. So as you know, there is what? Special types of sodium channels. What? Special types of sodium channel. So these sodium, it will stimulate what? The sodium channels. Again, there will be a huge influx of the sodium ion through these channels, and it will also move into this side. It will also move into this side, and there will be a huge influx of the sodium ions. Again, the in-plate potential will lead into what? Into threshold potential. And after threshold potential, if there is a huge influx of the sodium ions, then it will lead into depolarization. What? Depolarization. So we can say this depolarization or the leading of the in-plate potential into the depolarization or into the threshold potential depends on the influx of the sodium ions. If there is minute amount of the sodium influx into the intracellular environment of the muscles, there will be in-plate potential. Now, if these sodium go into the neighbor cells or next page area, and there will be if there is a huge influx of the sodium ions, then again the in-plate potential will lead into threshold potential, and after that there will be depolarization. So again, this depolarization, it will travel over the membrane of what? Membrane of muscles. So this is how the electrical impulses are like, we can say action potential transfer into what? Into chemical signals. We can say electrical signals transfer into chemical signals, and again chemical signals transfer into electrical signals, which goes over the membrane of the muscles. Now, when there is a contraction, or we can say when the action potential over the muscles membrane is over, when the action potential over the muscles membrane is over, then these neurotransmitter will be detached. These neurotransmitter will be detached from the what? From the receptor site. And what is the name of these channels? Nicotinic cholinergic receptor. What? Cholinergic receptor. Why this is called nicotinic cholinergic receptor? Because these ligands channels are sensitive to both acetylcholine and into nicotine. Therefore, this is called nicotinic cholinergic receptor. So, there is a special types of enzyme present over the membrane of the muscles. This is called acetylcholine esterase. Acetylcholine esterase, which break the neurotransmitter from the receptor site into acetyl and choline. Now, as we have already studied that acetate or acetyl, this is synthesized inside the neuron ending by the mitochondria. So there is no need to transfer the acetyl back into the nerve's ending. But the choline is absorbed from the GI tract. So the choline, it will again recaptured by what? Or retransferred into the neuron ending when the action potential is over. This neurotransmitter is breakdown by what? Choline acetyl transferase esterase. What will happen? The choline, it will move again into what? Into the nerve ending. But the acetyl, it will release into the blood, or we can say it will go into the liver. It will go into the liver. So this is what? This is neuromuscular junction. Now, there are some conditions in which there is muscle weakness, or we can say there is no action potential. There is no action potential. For example, we will study two disease or two syndromes. Like the first one is Myasthenia gravis. One point. the neurotransmitter released from the vesicle, these are broken down by what? By acetylcholine esterase. But there is a special types of proteins. What? But there is a special types of protein present over the membrane of the nerve ending, and this is called clathrins. What? Clathrins. And what is the function of clathrins? It will recapture the vesicle. It will recapture the vesicle into the inside of the neuron ending, so that it will be loaded again by a neurotransmitter. The neurotransmitter, the choline, it will transfer into the nerve ending. But there is also special types of protein which is called clathrins, which transfer the vesicle back into the nerve ending. Now there are two syndromes related to the neuromuscular junctions, are in which there is no nerve impulses formation, in which there is no contraction, in which there is no body movement. The first one is Myasthenia gravis, and the second one is Eaton-Lambert myasthenia syndrome. What are these diseases? Now, first one is Myasthenia gravis. In Myasthenia gravis, actually the muscles become weak. The muscles become weak. Why? Because there is no action potential. Or we can say there is no response of the body towards the stimulus. What caused the Myasthenia gravis? Myasthenia gravis, actually our own body immune headquarter, it produce antibodies. Which bind into what? Into nicotinic cholinergic receptor. So when the receptor site is already blocked by antibodies, as there will be neurotransmitter binding. When there is no neurotransmitter binding on the receptor site, is there any influx of the sodium ions? Is there any action potential? Then is there any contraction? Is there any movement? So Myasthenia gravis is what? Our body own immune headquarter produce antibodies which bind into the receptor site of nicotinic cholinergic receptor. Then the neurotransmitter are unable to bind into this receptor site. There will be no influx of the sodium ion, so there will be no depolarization, or we can say there will be no movement, or there will be no action potential. Now there are two types of drugs. Like one is pysostigmine, and other is neostigmine. What? Neostigmine. These two types of drugs are used in Myasthenia gravis syndrome. What is the function of these drugs? These drugs actually bind into the acetylcholine esterase enzyme. These drugs like pysostigmine and neostigmine, it will bind into acetylcholine esterase enzymes to block it. Now, as you know, acetylcholine esterase enzyme is blocked. Now, as there will be any breakdown of the neurotransmitter? Is there any breakdown of the neurotransmitter? No, because already the enzyme is blocked. They are deactivated by two drugs. Now there will be a huge or great accumulation of what? Neurotransmitter, and it will wait to find the available nicotinic cholinergic receptor. Now it will wait to find the other available nicotinic cholinergic receptors to bind, and again there will be action potential over the membrane of the muscles. So there will be movement. Another one is Eaton-Lambert syndrome. What? Eaton-Lambert syndrome. In Eaton-Lambert syndrome, there is also autoantibodies, and these antibodies will not bind into acetylcholine esterase, but these antibodies will bind into the calcium channels. It will bind into the calcium channels. Now, think, if antibodies bind into the calcium channels, is there any influx of the calcium ion? No. When there is no influx of the calcium ion, is there any release of the neurotransmitter?
No.
When there is no release of the neurotransmitter, is it will bind into the receptor site? No. There will be no action potential. We can say there will be no movement. So these two diseases actually what? It block the neuromuscular junction by one way, like by blocking what? Like by blocking the nicotinic cholinergic receptor, and other by blocking the calcium channels. This was something about the neuromuscular junction.